on top of my pile.
Thank you.
Good grade. Yeah, I didn't quite make it through all the labs. It takes...
No worries. Just want to make sure that
Yeah, for sure. All right, guess we're ready to get started. So here we are, week 10. So basically at the end, so just a couple of reminders. So again, I'll be around, until all the customers are gone, we'll say, today, right? So today's kind of the last day for whatever. I mean, it's not really the last day, but I'm just saying we have time set aside. So I'll be in our regular lab period. Again, let me know if you're coming late though, right? So I know to stick around. Otherwise, we can help with posters. If anyone needs to make up a lab, speak now or forever hold your peace. I don't think I have any lab makeups, but like I said, we could squeeze that in today if anybody is straggling. So I'll just put open office hour time. So like I said, let me know if you're coming, especially if you're coming later, so I know to expect you. And then, like we said, Thursday we'll have our poster presentation, or we'll call it our poster symposium. Since like we've talked about, it'll be more kind of poster session format, so less pressure as opposed to presenting in front of the class. And then if you brought posters, I can keep them in my office too, so you don't have to haul them around. So that looks good. And then, so other kind of announcements. So I'll take, we'll call it, or paper copies of assignments, ideally turned in by, so this Friday, June 6th, 5 p.m. So again, that's just when the mailbox closes. So this is ideally when the exam is due. Again, if you need more time, let me know, right? We can probably push this to like Monday over email, but I'd prefer that we just get them kind of done. And if you have questions, like I said, just reach out to me so you're not just letting it kind of linger. And so like I said, the paper, you can email it to me. And so that can be due by June 13th, midnight, we'll say. And then any other straggling assignments. So I've took a look at Blackboard, right? Don't forget your reading questions. So that could come back and haunt people. So work on that next week, since you'll have a little extra time, hopefully, with the take-home final. So don't forget your reading questions, and then any other assignments. So any other assignments you can turn in by email. So again, ideally by the deadline of June 13th, so I have plenty of time to grade.
So if those are like last minute labs, can we just take pictures of them?
Yeah, exactly. So yep, so just like the question, right? Last minute labs, just take some pictures, send them over email, and I'll accept them up to June 13th. And anything else that's straggling. But like I said, don't forget those reading questions. So get them done. Any other questions, comments? Basically at the end. And then of course I'll say bug me if you need help over the summer. I always look at my email. I'm a millennial. So feel free to email me if you need anything, like scholarships, whatever. You're just like trying to plan your life path and like what PhD programs are good. Like more than happy to kind of brainstorm or whatever else you need. More scholarships, internships, applications. So I will be in email contact. All right, let's dive in to our chapter then. So kind of, oh, sorry. You don't care about the candidates for VP. Just pulling up my notes. All right, so now we're going to finish up. And I do need to post the notes. I apologize. I've been slacking on that. So I'll get those going probably after class. And so we're going to be looking at polymers and our last two inner chapters. So we have inner chapters S and T formally. And so I'll just pull up our Macquarie inner chapters. So S is all about synthetic polymers. And so I'll bring in this demo. I'll save the demo for Wednesday when we have, or not Wednesday, Thursday when we have a little more time. And so this is a good reaction. So this is a pretty common polymerization reaction shown here in the beginning of your inner chapter. And so like I said, we can do a similar reaction. So I use slightly different reagents, but same sort of idea where we're going to look at these condensation reactions. And then T, so the next chapter will be DNA then, so kind of our biological polymers. And so of course there's nylon, and I mean something that we can kind of think about, not to go completely off topic, but of course as we discuss these polymers and what they are, so we have to think about the monomers that they come from, but then also how like these things contribute to microplastics and how they interact in the environment. And so that's kind of, again, a separate issue, but something to keep in mind as we talk about these polymerization reactions, how strong they are, and kind of what happens. And so what I just want to point out is, so of course nylon was one of the first synthetic fibers, and now we have all sorts of other synthetic materials, rayon, and all those though are synthetic polymers, and again essentially microplastics. And so let's formally define what a polymerization reaction is. And so our polymerization reaction is anytime we have repeated addition of small molecules, and this could be something like the same one, AAA, or maybe some sort of alternating AB pattern. And I think you kind of played with this in lab, at least with the structures, and then it's often kind of cartoonized, we'll say. So again, we have our monomers, which is then our individual unit. And so this is, again, whatever organic monomer. So we've looked at like ethylene, for example. So that would be our monomer. And then obviously repeating units of this monomer. And so you often see other analogies of like the train car and the train, especially if you're trying to reach out to younger audiences. And so then we can look at some of these mechanisms. And so there's a few different mechanisms by which polymerization occurs. So of course, a common one is this radical initiated mechanism. So let's add that. So this is radical initiated. And so this is the hydroxide radical. It could be kicked off by other radicals as well. So again, this starts to go into the rabbit hole of how you actually do polymerization on an industrial scale. But there's any other radical sources can kick this off. And so let's kind of look at our mechanism here. So I'm just redrawing our ethylene. And so then our little radical initiator comes along. So again, for our purposes, it doesn't matter. Then they're going to form this little bond here. So I'm trying to show half arrows. So our radical is contributing 1 electron to our bond. So we have something like this, but then, right, so our double bond, 1 electron went to form a bond with the radical initiator, and then the other electron is now a radical, so as shown here. And so now this can propagate with another ethylene, and so we can again appreciate how we just had 1 radical initiator to kick off our reaction, and then it'll keep kind of going right in this radical mechanism. And so that repeats, and so of course you can see that kind of shown here again with this hydroxyl radical initiator, and then we end up with our radical. Again, they didn't draw it out, so just kind of shorthand of this, and then again continuing on and on and on. And so then this continues. So again, this is kind of where we're not going to get into the engineering of it, obviously. So that's like an entire separate course, and even that is only just a taste. But depending on, you know, your concentration conditions and so forth, you can kind of control the chain length. So that's kind of what you want to be able to do. So you want your polymers to roughly be, right, again, the same length, so they have similar properties, and then you can continue to do whatever with them. And then many of these radicals terminate by this termination reaction. And so this just kind of tends to happen. So like I said, that's where controlling kind of the concentration helps control when this happens. And so of course, if you have 2 radicals, then, so it doesn't matter what's attached over here, right? Could be the R, could just be more ethylenes. And so now if we have 2 radicals come in contact here, so that ends our polymerization. So right then they form their bond here, and now there's no more radical, as 2 radicals again came together, so there's nothing to keep this reaction going. And then, so again, we've kind of seen this, so we want to be able to translate our monomer unit then to then our repeating polymer, and noting, right, that we're breaking the double bond, or so in this case, right, we're breaking the double bond. We'll look at other types of polymerization reactions, mainly the condensation one, and we'll see how that is a little bit different. So again, break our double bond for this kind of radical initiated mechanism, make our single bond here, and then obviously chain and chain. And then you have N, and so again, that's where you indicate how long your typical chain length is. And again, right, that's what you would be, if you wanted to be a polymer chemist, that is what you would all be focused on, is how to control this chain length and, you know, in whatever pattern that was desired. And so then there's obviously a lot of famous materials that have come out of the development of all these different polymers. And so I'm just highlighting Teflon. So of course Teflon, you have a bunch of fluorines instead of just hydrogens. And then polytetrafluoroethylene here is the proper name for, again, Teflon coating. Not that I'm a fan of Teflon coating personally. And so other cool, notable polymers are So I couldn't find like a perfect comprehensive page. So right, try to feature some female scientists from time to time. Obviously, the field has been historically male dominated. That being said, a lot of times, their wives acted as assistants, but she was obviously an early polymer research chemist at DuPont. And so let me pull up another page though. I just wanted to highlight her biography, but let's actually look at Kevlar, the polymer. And so they were just exploring different polymers. And she was working again at DuPont, noticed some polymers, made some pretty strong fibers. So then she tested them and found that they were pretty strong. And so here's the structure of Kevlar. And for those material-minded people that might be interested in this, so it mentions that it has high tensile strength, so it can withstand being stretched. So again, that's why it makes it a good material for bulletproof vests. So that's what most people are familiar with the application of. But of course, low compressive strength, so obviously, right, not good for building materials or anything with structural integrity. So just kind of cool, because Kevlar was, again, basically an accident, just kind of playing around with different polymers. Oh, sorry, printout went on another sheet. And so again, we're not going to memorize any polymers and monomers. You do, again, want to be able to figure out how they chain up. So like with polypropylene, you're going to notice it's going to have a methyl group hanging off, but it's going to chain up, right, very similar to ethylene. And so this is just stolen from your book. And these are also kind of more, I'll just say, like traditional polymers. Obviously, right, I don't know if anybody did their paper on it, but there's all sorts of cutting edge. Like now they're all into biodegradable polymers, and like I know they're trying different, what are called like polylactic acid based polymers, PLAs, for 3D printing. If anybody does 3D printing, so you might be more aware of that. So things that kind of degrade like when exposed to water. And so again, right, that's like the beauty of these polymers, but also why now all the research is going towards like biodegradable. And so we can see what they're made in. So polyethylene is used in a whole bunch of things, obviously, right? Shrink wrap, bags, toys, everything is made out of plastic. You guys know that. Same thing, so polypropylene, again, very common. Food packaging, clothing, again, more plastic parts, like we can find plastic everywhere. But again, right, and so now we've made these really strong bonds, and so the problem with these bonds is now, again, they're hard to break down. And so that's where, again, the whole microplastic sensation, which, I mean, I think there is some concern there. Obviously, who knows how much. But if these materials can't be broken down, so the microplastics are just broken down physically and not chemically, since again, these bonds take a long time to degrade. And so some of them, you can degrade these polymers with like UV, but then how do we normally bury our trash? Is it exposed to light? So no, so then it's like, well, we've buried it, so now who knows how long it's going to sit there not being exposed to UV or anything. And so again, just kind of looking at other polymers while we think about the environment. So PVC, so that has a chlorine group on there, so again, very similar to ethylene. So again, it's going to chain very similarly. Neoprene, I like neoprene. Like I mentioned, this is one of the options in case you are allergic to nitrile, which is very rare, but like I said, does happen. And I want to say, like, I think it's the skin condoms are made of neoprene, so they're also, if I remember correctly, so slightly different material. So again, if you have allergy needs. So that's cool how it chains up. So this one's a little bit different. You can kind of think about the mechanism. So kind of what happens is these double bonds shift over. So probably you have a bond formed, and then this gets broken, shifting our double bond over, and then kind of propagates there. So that's how you get your double bond in the center, in the polymer, but two on the side there. So just kind of moving your electrons down the line. And then we have plexiglass. So methacrylate is our monomer there. So this is obviously used a lot for visors and everywhere you need, right, some sort of see-through plastic. And that's always hard to accomplish, is getting anything like clear in plastic manufacturing. So that's like a premium. And again, it's going to chain at our double bond. So we saw Teflon, like I said, with those fluorine groups. Acrylon, so I'm not too familiar with this, but obviously it has just a different cyanide group here, or nitrile group. So again, right, we can find it in everywhere, a bunch of synthetic fabrics. And then styrofoam. So this, I think a lot of people are obviously familiar with styrofoam, but we can look at the structure and then answer why styrofoam is a pain to recycle, aka pretty much nobody accepts styrofoam, which is kind of unfortunate. But this little styrene group here is, like I said, makes it not good to recycle, so now it doesn't work. So obviously, like all of the high density polyethylenes, you can like kind of re-blow them, like low density, high density. But then styrofoam with the different group there, it makes it not good to mix. So unfortunately, that's why a lot of facilities don't take styrofoam. And obviously we know it's used in packaging. Again, I'm sure you all have seen it. And so again, right, y'all are hopefully future engineers, so things to think about. So not only, right, like what polymers to use, how to make packaging, like use less plastic, and I'm sure you guys see that just when you buy stuff at the stores, right? They're always like trying to minimize the plastic amount, and obviously there's structural concerns with that, so always trying to balance those factors. And again, getting into biodegradability now too is the new frontier. Oh, and something we also should have mentioned, I forgot, just to point out, I don't know if this is obvious or not to y'all, but they're made from crude oil, right? So that's the other thing. I don't know if you noticed that in like the media. They're always like, oh my gosh, it was made from hydrocarbons or gasoline or something. But I mean, that's just like where your organic monomers have to come from, right? Like that's a good source of just carbon basic building blocks. So I never know why that gets all like crazy sensationalized. Like, oh my gosh, they are fossil fuel products because they're organic. Anyways. That's why you don't listen to mainstream media and you read papers. So now let's look at our condensation reaction mechanism. And so let's just point out some of our groups here. And so what was this group? You remember? group is this one? It's one of our common ones. Should remind you of maybe titrations or something. No guesses? So remember, this is our carboxylic acid group. So we can recognize, right, our acidic proton. So always good to kind of recognize that. Again, just kind of preparing for OCHEM, hopefully. And then we have, do you remember what our NH group is called, our functional group? What are these guys called? I was gonna think of like Breaking Bad too. So amines, I don't know, I always think of methylamine for some reason from Breaking Bad. So that's methylamine. Again, one of the many ways to make meth. All right, so then looking at this condensation reaction, so we kind of have it color-coded, but we'll go through the mechanism. And so what we're kind of seeing, so as right the name implies, so condensation reaction, we had discussed that that's when we're going to lose water. And so we're losing water, and this also means that we're forming a bond though, because again we'll look at hydrolysis, which is basically the opposite. And so then we can look at our mechanism. So right, they're color-coded. We're going to kick off this OH and then form what's called our amide bond here. So this is a carbon-nitrogen bond. And so let's go through our mechanism roughly of how this is going to happen. So we have our carboxylic acid group here, R. And so then we have our amine. And so, right, these amines have electrons there, which can attack our carbonyl group. So that's what's going to happen first. Sorry, just trying to abbreviate a little bit so I don't have to draw the whole polymer. So then we can attack our carbon here. So we've mentioned that this can be considered an electrophile, so meaning, right, it wants electrons, and these can move up. All right, so now let's draw our step here. So again, negative charge here, we've made our bond. So H, H, R, so positive charge here. And so there's probably a proton transfer mechanism. So right, we have this OH group hanging right next to this hydrogen, and so that's not happy. So I'll go proton transfer, or right, protonation, deprotonation. So like I said, I'll let your OCHEM professors kind of get into whether it's a proton transfer or water aqueous back and forth mechanism. Again, they'll have strong opinions. So then we can have water. So water is ready to leave. We still have our, what was our double bond, right, hanging over here. NH. So now it's neutral. And so then we can reform our double bond. And now water is ready to leave. And so now we lose water. And then we're left back with our double bond. And now our amide group there. And then of course, right, R and R for the rest of the structure. So that kind of makes sense for the mechanism. So it's at least good to kind of work through it. Like I said, I won't expect you to do this on an exam. But once you get to OCHEM, you really do kind of want to understand these mechanisms so that you really understand how everything comes together, which again takes time and practice. So this is a reaction that, like I said, I'll do it tomorrow. Or not tomorrow, Thursday. I keep saying my day's all wrong. And so we can appreciate how these chain up. And so let's go through this one. And so this is called our nylon synthesis. And so more specifically, this is called nylon 6,6 synthesis. And so the reason it's called 6,6 is because both of these pieces have 6 carbons. So 1, 2, 3, 4, 5, 6. And so there we get, so also note the 2 pieces. So we want to do a condensation reaction. So right, we can pick 2 reagents that will do that. So this is again, right, a diamino. So 2 amines, right? That makes sense. Okay, so we have 2 amines to chain up. And then this one, again, 6 carbons. 1, 2, 3, 4. 1, 2, 3, 4, 5, 6. So carbon to carbon. And so again, 6. And then pointing out, right, that again, they are carboxylic acid. So on both ends. So right now we're seeing, okay, we can chain up kind of here to here. And so that's also kind of something to map out in your mind. So, okay, these can match up, and then obviously, right, another amine here will come. So, okay, we're just appreciating where they're going to go, because again, this is just going to be an ongoing polymerization reaction. And so again, similar mechanism, so we don't necessarily have to go through it, but just kind of right, you have your nitrogen, lone pairs aren't drawn in there, but you need to be thinking about them. They'll attack their carbon center again. This goes up. Again, that's a pretty common OCHEM mechanism. And so skills you want to have is be able to identify your amide bond. So if you've taken a look at your exam, right, that's something that you kind of have to do with some of those polymer questions or DNA questions, is like, is the linkage correct? And so you just want to examine that nitrogen carbon amide bond. And again, right, was it linked up appropriately? Is the R group drawn on there all correct? All those sorts of things. So that's what you're looking for on your exam. And then this is obviously showing us our monomer of repeating units, but right, it's made up of 2 different monomers here. So repeating unit made up of our 2 polymers, so our copolymer there. Oh, and so right on cue, so copolymer. So just definition, right, polymer made up from more than 1 type of monomer. So again, and like I said, we're mainly just taking a quick look at this, but obviously you could also have like 3 copolymers. That would be more rare. What you usually see is like a bunch of systems of 2 polymers, and then people will try to kind of mix those 2. And so I think we've looked at this, but we'll just take another look at this mechanism. So again, this is our esterification reaction, and we're just pointing out that it goes through a very similar mechanism. And so instead of an amide, we have an alcohol. So again, this is our synthesis. Very similar, right, to, again, our ester synthesis is what we can kind of compare it to for making the nice smelly things. But if you have, right, a polymer here, so you can make it an esterification polymer by having your carboxylic acid on both sides. And so I think that's a trend hopefully you're picking up on too, right? It's like, so this one, if we want this polymerization reaction to go and go and go, it has to have an alcohol on both sides and a carboxylic acid, right? So it attaches on both ends. So that's kind of different from the basic synthesis. So let's compare, right? So let's say compare to ester synthesis, where it's just, right, one alcohol group and typically paired with, again, so 1, 2, 3 with our carboxylic acid. And so, right, again, it only has one alcohol, one carboxylic acid, and so we can only make our one structure. And so again, we can go through the same mechanism, or very similar, and appreciate this. So our oxygen, lo and behold, it's going to attack that carbonyl. Who's surprised? And so then let me draw it out. So electrons go up. Sorry, making sure I don't miscount my carbons. 1, 2, 3. Still have our OH attached. And now we have our other alcohol group. And so again, right, positive charge. So I'll be lazy and call it a proton transfer, because again, I'm an organometallic chemist, not an O chemist by training. So then water, got our alcohol group, reform our double bond, eliminating water, and then, oh, so 1, 2, 3, and then our alcohol group there. So that was our, again, ester synthesis mechanism, which again came up on the exam, so I'm trying to give you a few exam hints, right? So this is now related to your alcohol, and so that part, right, is from whatever your carboxylic acid was. And so again, comparing the polymerization of an ester, it just has to have it on both sides, so obviously we can continue on and on and on for however, right, we have set our conditions to. So a lot of, like I said, a lot more engineering goes into polymerization when you really get into it. So I'll just kind of point out 2 alcohols, 2 carboxylic acids, so it has somewhere to attach. And so just kind of, again, more fun facts from your book. And this is probably getting old, so I should have updated it. Basically, we use a lot of plastics. I think we all know that. Again, I don't know. That's the one of the few things that does keep me up at night sometimes are single-use plastics and just their prevalence in the environment. And so, like I said, this is from your book, which I believe was published 2018, 2019 or so, if I remember correctly. So I'm sure these figures have only gone up. So especially like in the Asian countries, that's very popular, the single-use packaging, you know, everybody uses plastics everywhere. So again, not sure what we can say about that. Hopefully y'all save the future with biodegradable plastics. And so then other sort of reactions are called these like cross-linkage reactions. And so these are where you can have these two sulfur bonds, and then you make repairs across this, so across these disulfide bonds. But what's kind of cool about this is, so there's a real world application that some of you, especially ladies, may be familiar with. I don't know if you guys have heard of Olaplex, or I'm bad at pronouncing it. Olaplex? No, maybe. Usually your hairstylist is like trying to pimp you out on this or like treat your hair with it. But I looked into it, and there is actually science behind it, and the science behind it is that it repairs the disulfide bonds of your hair, so it really is supposed to help with like damage. So it seems pretty cool, seems scientifically backed, so thought I'd point that out. Again, right, lots of careers to think about in cosmetics formulations too, so kind of non-traditional careers. I feel like we're going through this chapter super fast. All right, let's start looking at proteins. Like I said, hopefully we don't blow through these chapters too quickly. And so, of course, proteins are now considered biological polymers, essentially. And so our monomer would be our amino acid. And so I'll probably print this out during the break so you guys can have this sheet. So this is from Compound CHEM. So again, they always have really nice drawings and graphics and stuff. So I stole this from them so we can look at it. And so a couple of things to look for. So of course we have our basic structure, and so we can already maybe tease out how they're going to chain up with one another. So we have an amine again, surprise surprise, and then a carboxylic acid again, and then all amino acids have the same structure. And then they have a hydrogen and then some R group, except for, so the only exception is glycine here. So glycine just has another hydrogen. And so glycine is not chiral, but it's the only exception. And so all these others are have what's considered chirality. So right, this carbon here is going to be a chiral center. And so right, refresh yourself what that means. So it has 4 different things attached. And then it has right-handedness to it, so stereochemistry does matter. And of course, we have natural amino acids. We've talked about the chirality of life in here, right? Or no? I don't remember. I feel like we have, but... Yes, no. Remind me if we didn't. Because sometimes I show this like paper where it talks about the chiral center flipping.
I think we received that.
Okay, so yeah, I feel like we've talked about it, but I wanted to make sure that we at least touched on that, because part of, again, the chirality argument is you can deprotonate this carbon here with the base. Yeah, I always like ignored in paper, chirality of life. Sometimes I could have talked about it like first quarter too. I don't really remember. If not, I can try to find the paper for after the break or for Thursday. And so again, just kind of looking at our chiral center. So right there's our carbon. And so that's why these are drawn with wedges and dashes, right? Because again, chirality does matter. So chiral center, right? So all these matter. So again, just kind of appreciating them. And so then, again, I'm not going to make you memorize any amino acids. I can't remember. Some biochemistry classes sometimes make you memorize parts of these, which seems terrible, but you can at least kind of parse out some of the structures, right? Like phenylalanine, so that has a phenyl group at least. Tryptophan, so I just point out tryptophan because it's pretty common or is basis for a lot of drugs, including recreational drugs. So let's see, you got isoleucine, leucine. Trying to point out any other cool ones. Some of them are charged. So again, just kind of to appreciate right now, like aspartic acid, so there's a charge on it. Glutamic acid, so they are going to be pH sensitive. So I'm actually not sure what pH they're shown at anyways with this structure, but all that means is, right, that they can lose protons or gain protons, right? So they're very sensitive to the pH of their environment. Methionine is kind of interesting. Cats require methionine sometimes. Well, they need it, so you might have to supplement like their food with it or essential amino acids. So cats can't synthesize all their amino acids. Let's see, cysteine. So again, just kind of interesting to point out the ones with sulfur groups as well. All right, that's probably a good spot. So let's take a break there. So I'll go print out this so you can have some reference material. And maybe I'll see if I can find that chirality paper. let me. All right, like I said, let's continue. Hopefully we don't move too fast. So I did find the paper, and then y'all can tell me if we've seen this paper. Oh yeah, so we have, I'll at least just give, again, the quick, we won't get into the philosophical argument unless you want to, but I'll point out, again, just the base and the chiral center. So right, there's a hydrogen there that's not currently shown. So again, organic chemist, right? You have to fill in your hydrogens. And so that hydrogen there is susceptible to deprotonation by that base, DBU. That's just a large, well, it's considered a larger base, so it's kind of softer. So it's used a lot in organic chemistry. And then looks like protonated solvent or like some sort of polar solvent to get the reaction going. And so again, you can flip the chirality. And so I won't go too much into it if I've already talked about it, but I just wanted to bring up the paper again and kind of share it, since this is, the paper is from 2008, the one that I referenced just because I like the pictures, but this is still a topic of research and conversation, of course. All right, we're gonna fly through this chapter. That's okay. So let's just keep going for now. So like we said, we pointed out our chiral centers. So we have D-alanine and L-alanine shown here. And so life is L-based. And again, right, has handedness. And then anything that's called a racemic mixture is considered like 50-50. So, right, it's going to be both of them, so D and L, or R and S, depending on how you're naming your stereocenter. And a lot of synthetic, so like for research purposes, when you make like peptides and stuff, you work with the D ones. But I think I've mentioned it before in this class that some people, like some researchers, are coming up allergic to the unnatural amino acid. So just kind of anecdotal tales from researchers. And so like I had hinted, so we're going to see similar bond formation. And so that's something that you'll practice or look at, like I said, on the exam. That's obviously an exam question. Is the amino acid chain formed correctly? And so let's look at a couple of these. And so back when I used to actually do peptide chemistry for a short period until I decided I did not like peptide chemistry. So peptide chemistry, all these reactions, a lot of them use piperidine as one of their reagents. I believe it's used as a base. Let's look it up. Piperidine. And it smells a little horrible. I don't know. Once you work with chemicals long enough, you can kind of smell who's toxic. And so this one is not fun to work with, and I believe it is a teratogen. I was trying to find the official labeling. So it is toxic, and it mentions it has this weird pungent odor. Blah, blah, blah. But yeah, it's not the greatest reagent. So obviously used a lot in DNA synthesis, peptide chemistry. Yeah, they don't have it on there. But it's not the greatest chemical to work with. And like I said, it smells. But a lot of these, so it's actually really cool what these things are used for. So some of the research is going to be for like picomolar detection, or probably even smaller potentially, but picomolar detection of, let's say, cancer. So that was specifically the research that I was doing. But obviously, right, you can try to detect whatever you're interested in detecting. And so kind of the idea is with these peptide synthesis that they try to use to detect cancers is you have this little, they call it a little bead, but it has some sort of like activator, so it glows, or not glows, but it turns like purple when it's bound to the cancer, so when you have like a whole thing bound. And so then you just go in order of whatever your peptide chain that you desire is, so etc. Although what's cool and like what people try to do now for like large scale peptide screening of these is you really want to make a library. So back in my day I was doing this by hand, and you just like keep remixing all the possible combinations, so you get like basically a randomized library of different peptide synthesis. But now they're trying to outsource that to machines so that the machines can pump out all sorts of peptides much faster, and then they can screen them obviously against different cancer markers, or again whatever you'd want to apply them to now. And so again, we're appreciating kind of order doesn't matter, right? It's just whatever is being called for to code. So obviously we can start talking about the primary structure too. So that's formally defined on the next slide, but we might as well state it now. So our primary structure is just our sequence. So again, we're at least just getting a preview of biochemistry. And then obviously biochemistry is like a huge field in and of itself, and it depends kind of if you want to go more bio focus, more chem focus, and how you want to take it, right? So I've kind of mentioned, like me personally, I'm always caring about the active site if we're looking at something biological. Other chemists, right, are going to be really looking at the primary structures and maybe those secondary, tertiary structures, which we'll get into, and like figuring out what causes diseases, like if that's misfolding, right, in the secondary or tertiary structure and so forth. And so again, key points, peptide bond formation is just another condensation reaction. Again, so okay, right there are our pieces. So carboxylic acid pairs with our amine, just like before. And so again, right, we just have to pick the appropriate order. And we're saying like, hey, make a peptide or an amino acid with these amino acids. And so then, right, obviously they'll have very similar structures, and I would set yourself up for success of like, look at this very general form. So this is, and so all that's changing and all you're really looking at is what is the R group that's attached to that carbon there. And then you can figure out, right, what your correct structure is. And so again, just kind of draw it in whatever way makes sense. So you can clearly see that amide bond formation and, right, determine which amino acid is which. So, right, amino acid 1 here, number 2, etc. So again, just kind of identifying where those linkages are. Obviously, we're losing water. Again, I think that was expected since it was a carboxylic acid and a hydrogen. And so again, right, set yourself up for success. Who cares about the rest of the structure, really? So R group, and then, right, general form. So then we can R group, and again, just kind of keep going. So just watch those key linkages. And so same thing. So other things, so all we did was show alanine and serine. So obviously alanine first, serine second. Again, we could flip the order if they had said so, right? So serine first now, alanine second. Just looking at their bond formation there. And so again, I like this kind of general template that y'all should reference probably to help you. So again, right, looking at your R groups, and I would maybe point out your amide bonds. So okay, amide bond, so you can see where every new amino acid is. So that should help you with your exam portion as well. So I was trying to find some good pictures. And so let's just look at our primary structures first. So again, like we mentioned, this is just our sequence of amino acids. And I mean, at least cool to appreciate how many amino acids are in all these different things. So of course, insulin is 51. Insulin was pretty, obviously, a very miracle drug. And then snake toxins, so a little low at 62. I mean, again, this is just, we're just comparing. So myoglobin, right, so the main structure, 153. So keratin, 204. Hemoglobin, so 574. And right, looking at collagen, so again 3,000. So you can just kind of appreciate our primary structure. And so then I was trying to get some good, it's hard to get good pictures of proteins and stuff, admittedly. You always just get these oversimplified drawings. And so I was trying to find, like I said, good pictures of our secondary structures here. Oh, so here's at least some of our secondary structures. So the main two ones that you see are this alpha helix kind of coily structure here, and then this beta pleated sheet. But you could also have technically just like random coils or triple helixes, or again, you might find other not as common secondary structures. And so, like I said, I was trying to find some good papers that kind of get into this. And so this one at least gives you some sense of what they're going to be doing with the secondary protein structure. And so the secondary protein structure, I highlighted this key part here so we can understand what we're looking at, right? Because each step is supposed to be kind of like different interactions and different complications. And so the secondary structure starts from our folding from our hydrogen bonds, right? So however it's starting to hydrogen bond within itself. So that was kind of cool. And so, right, they get coiled and folded in, again, a certain pattern depending upon the hydrogen bonding. And so, again, it mentions, right, the two main structures are the alpha helix and beta pleated. Like I said, you can find all sorts of other not as common secondary structures. And let's see. So blah, 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 beta sheet. And so I wanted to point out, again, some of the research. Like I said, I didn't think we would get so far today, but we're kind of going through this chapter. But basically, so the research areas and where does the misfolding occur. And so it mentions here that the secondary structure of our protein can, of course, be altered by mutations in the primary sequence. So, that starts to get into like damage to the DNA, and then what does that lead to? Well, it leads to a miscoding, so then you get something wrong. And so, right, then you get your mutation. And so obviously any sort of mutation will start to affect all of these structures. So again, we're just trying to focus on the secondary. And so it mentions like prion diseases. I'm not sure what this is. Spongiform encephalopathies. This sounds like some sort of brain swelling. Again, the more biology-minded people can help me. And amyloidosis. So again, all these are different diseases that arise from the secondary structure. So that's kind of cool. And so, right, again, it goes on to mention that messes up the folding of your beta sheets, and then, of course, that can lead to tissue damage. And so it mentions sickle cells, so glutamic amino acid is replaced with valine, and so again, that leads to misfolding already, right, in the secondary structure. Oh, so this is Creutzfeldt-Jacobs. So I vaguely remember this, but like I said, I'm not a biologist, obviously, but you can appreciate where the research goes with this. And so again, right, this is all to say that any sort of little mutation in just our amino acid sequence starts to lead to a chain of issues, right? Obviously starting with the secondary structure there. So that's kind of cool. And so then our tertiary structure, of course, starts to get into what the whole 3D shape of the protein is. So let's just kind of pull up a few. So of course we looked at, mentioned hemoglobin, and so of course getting into the big structure. And so you can kind of see the different folding units. So, right, your secondary structures, your alpha helixes, and then they come together to make your overall protein structure. And let's see, I thought, did I leave off quaternary? And so quaternary, we'll add that. And so quaternary, if I could spell, so that gets into interactions of usually two proteins is what it is typically defined as. So then, right, if your protein interacts with another protein in some manner, then that gets into the quaternary structure. And so I'll pull up the chapter in your book too. So where I was hoping they would show better pictures. This is why. Well, I mean, we can also talk about the limitations of it. So obviously it's very difficult to get crystal structures too of these large biological proteins. They can be done, so that's really cool research, but that is, again, how you get these X-ray structures. So, right, we've talked a lot about coordination chemistry, and so we've mentioned that you have to get a crystal structure. And then, I mean, you guys have at least made like aspirin crystals in lab, so you've seen that. But you really need like a nice, good crystal for X-ray diffraction. And that's hard enough to do with these coordination compounds and, you know, even slightly more complicated metal complexes. But you can imagine how difficult it could be to get a crystal structure of these different protein structures or any sort of data. So just looking at this X-ray in your book is pretty fascinating. And so, like I said, this could be a year's process of like a single group trying to figure out how to crystallize out a protein and get our active site structure. So again, obviously I care about the active site. And so this is a good, again, kind of picture too, so we can appreciate our secondary structures. So again, it's trying to show the hydrogen bonding here, which is starting to lead to the correct folding, right? So again, either perhaps alpha helix, beta, or the other ones you might encounter. Let's see, we might come back to the protein structures if I can get better pictures. And so then we kind of get into DNA synthesis, which I might save some for Thursday too. So of course we have DNA and RNA, and so we can look at our basic structures. So we have our nucleotides, and so it's made up of a sugar, a phosphate group, and then some sort of base. And so we have our 5 bases that occur. And so we have adenine, guanine, cytosine, thymine, and uracil. And then, again, you may remember from biology or that thymine is only found in DNA and uracil is only found in RNA. So whatever you need to do to remind yourself of that. So let's see, what other things should we point out? Let's see, we have our sugar. And so then we can see how our synthesis occurs. So building up our DNA chain. So again, just kind of identifying our pieces. So that's our phosphate backbone here, sugar, and then it's attached to our base. And then it just continues, right, with this sequence. So again, another phosphate base, sugar, base. So this is just building up, right, 1/2 of our DNA ladder as well, right? So this is just forming, so I guess we should back up a little. So we want to say, right, that DNA is, of course, this classic double helix structure. Let's actually see if the book had a good picture. Although I think most people are familiar with the double helix, but so of course, right, we have our double helix, so kind of a ladder that's kind of twisted. And so what we're looking at over here is just the synthesis of one side of that ladder here. And so then we form our double helix. And so if you remember, right, you can figure out who's paired with whom, or at least now this kind of explains it, why certain ones can only pair with one another. And so if we look at our double helix, we have our hydrogen bond forming across. But then, so now let's compare which are appropriate base pairs. So if you remember, I mean, again, I don't know if you've taken biology or not, but usually, so I always think of like straight letters with straight letters, and then of course U, and then right, C and G are both kind of rounded looking, so they pair together. So okay, that's great to memorize, but let's look at why that is now. And so we can see, okay, well, A and T have to pair together because they have these two places where they can form two hydrogen bonds. Okay, so they're hydrogen bonding there, so that allows them to pair appropriately. Versus the C and the G ones, so now they can form 3 hydrogen bonds across. All right, so that's where, like I said, we all kind of memorize that, at least in high school or something, and then we don't know why. So that's why. So then, of course, now our two sides of the ladder get to come together, and it has to be paired with the appropriate base pair. So again, like we're seeing A with T, or if we were looking at RNA U and C and G together. And so yay, now we can look at our backbone here. And again, just appreciate, okay, T and A, C and G. So nothing too crazy there. And so then, I feel like we're going to finish this chapter. Well, we probably won't finish the sugars, but let's see how far we get in the DNA. And again, y'all can probably help me, or you might know this. So now we can say, well, how do we code? So now getting into, right, our synthesis of amino acids. So how do we code for different amino acids? And so, of course, we're answering our question. So we have our DNA. And so we have our 5' strand, and so let's say it's ACC, TCG, AG there. So 3. And then the complementary strand is then, right, 3', and then just match all the opposite letters. So A with T, C with G, G, A, G, C, T, C, and then 5'. So it's opposite, right? So this goes 5' to 3', and 3' to 5'. And so then we can see kind of what matches up. So if we go back to our reference sheet here with our compound chem, so it lists here, I thought, yeah, I should have it on another page of our notes here. So this is one reference guide for all the different ways to code for this amino acid. So all these different sequences. Or, like I said, I think it's further in the notes here. So page 9 also gives us kind of a reference. So again, right, you start center and then code out so you can figure out what amino acid we're calling for. All right, so that's answering, right, our DNA. So our DNA tells us what amino acid we're doing, and then again, obviously, transcription errors lead to mutations. So let's see. So we'll keep going with our starches. So we might end up finishing lecture, and then we'll probably review or something on Thursday, or can look at other examples. So then starches are essentially just polymers of sugar. And again, is anybody surprised that this is another condensation reaction for these to come together? So condensation again. So very common theme in this last chapter. And so then we can look at our structures of glucose and fructose. And so some things to point out is glucose is a 6-membered ring, 1, 2, 3, 4, 5, 6. Fructose is 5, 1, 2, 3, 4, 5. And then with all these sugars, kind of their orientation and their chirality does matter. I always say that's why I'm not a sugar chemist. And so then, again, they form here. And then, so what's interesting about sugars is then, again, how the angle at which they interact determines kind of what's called the form. So then you have your alpha form, where you have this OH on the same side of the ring, so they're both kind of pointing down. And then you could say, right, that this formed alpha. So this is your alpha bond here. Again, right, just referring to the angle. Versus if they're on opposite sides, so this is down, this is up formally for our alcohols, and this would be our beta form for our connection. So again, that's something that you have to pay attention with sugars, is all these up downs of the OH in their ring. see. Trying to look at your homework too. So yeah, there's not too much. I was just trying to make sure you don't need to draw like overcomplicated starches and worry about these alpha beta connections, because like I said, that does matter, but I don't think we need to go into it in this class. So let's see, let's skip coding for now. We can go back to coding. So this is from your homework though, so we will look at this. And so your homework asks you to look at glucose and galactose. And so I went ahead and at least put them out here for us, so just copying our picture. And so kind of take a moment and see if you can figure out kind of what's the difference between them, because it's pretty subtle, but like I said, these sugars, you have to pay attention to the up-downness of those alcohols. So what do we notice about them? Do we see a difference? It's pretty subtle.
The H and OH are 4 are swapped on galactose.
Nice. So this is literally the only difference. Again, I think this is why I despise sugar chemistry. But yeah, that is literally the only difference, is that this OH is down over here in glucose, up in galactose. And so then, okay, so your homework asks, are they identical? So, okay, they're not identical. And so I like how you phrased it, the difference at carbon 4 here. And then I don't know if y'all know the name for this. So they're called diastereomers. So it's called a diastereomer because they're only different at one chiral center. So again, you'll get into it with CHEM more, but enantiomers are just like straight mirror images. But right, this is not a perfect mirror image because everything else lines up. So then, yeah, going through the homework, and so yes, they are isomers. And again, I defined it as diastereomers, which we've kind of touched on that term in here, but like I said, you'll really get into it more in OChem because we don't really look at a bunch of chiral centers, so obviously we wouldn't talk about it. And so optical isomers, so yes. So you should be able to distinguish it potentially by the difference in rotation. Again, with the difference in chirality. And then it asks if they're enantiomers. But again, right, I think this is always a difficult concept sometimes for OChem students. But again, enantiomers would be if every stereocenter was flipped. so complete mirror image, versus the diastereomers is, again, you'll get like a mix, like where one is the same and one is different for the stereo centers. So a diastereomer requires multiple stereo centers as well, or an enantiomer obviously can just have one. So I think, like I said, we'll save the other thoughts on DNA. So we'll practice this, I think, on Thursday / I might try to find some good videos. I want to say it's either Professor Dave or somebody else has a really good DNA transcriptase video. And of course they have cute little pictures. And then, oh, the other thing we should maybe look at for your homework is, because it asks you on number 3 to do alpha D glucose versus beta D glucose. And so let's again look at that. So that goes back, so let's find our notes. So right, we have our alpha and beta form. So again, alpha same side of the ring, beta opposite sides. And then I'm just trying to help you approach these things too. So opposite, if I could spell. And so then, right, I would kind of approach this. You might have to look up glucose. I wouldn't expect you to know it. And then something else, maybe we'll talk more about this on Thursday. But again, I'm like, this is why I'm not a sugar chemist. So sugars can be represented in all these different forms, and they all mean the same thing. And so we have just kind of this skeletal form of glucose, but of course it doesn't show you kind of the ring structure here. So here's what's called a Hayworth projection. So again, it kind of simplifies this up-downness. And then what's also confusing is a lot of the time you see what's called this Fisher projection, and they have our carbons kind of labeled. So that is again just another representation of the same thing, but we might take a better look at that on Thursday on how to translate it. And so these projections are kind of honestly difficult to work with, so I don't like them. So let's find a better picture, to help you. So that's, again, that's something that I'm just trying to point out with the sugar chemistry, is you can find a lot of pictures that aren't necessarily too helpful. So this one might be our best one here to kind of help us through it. And so again, then we want to compare. And so this is where it gets confusing, because we want to make sure that we're looking at our OHs, right, relative to our oxygen here. So our oxygen in the ring. So again, opposite sides versus same sides. So then, right, kind of looking at it, okay, now we can, right, orient ourselves. And then now we can understand that this is alpha D glucose, right? So we're looking at this OH and this OH. Again, I'm just basing the position off of this oxygen. And so they're both down. And so they're, again, on the same side there. And so let's just kind of copy that as a reference, right, for alpha 1. I'll put it off to the side. And so I'll just kind of copy that. So our 6-membered ring. And again, showing our down position. CH2OH here, so our alcohol group. And then, right, this is up, this is down. Make sure I drew that correctly. And so again, I'll just draw this as our alpha D, highlighting which ones are down. And so then we just have to flip one of them, right, to make it beta. So I'm going to flip this so it's opposite for our beta. So now this goes up, and then everything else is the same. So again, looking at those relative positions there. And so now they're opposite, so beta D glucose. Questions? So like I said, we'll hit back DNA, so we'll get more kind of coding with it. So again, sugars, try not to get too hung up on it in our class. Like I said, we'll probably look at more kind of Fisher and Hawthorne projections, just so you can at least kind of be prepared for the future, because like I said, that is a difficult part of sugar chemistry, is like trying to translate these flat structures to the ring structures and back and forth. And I mean, I'll admit I'm not that great at it, but that's why you should expose yourself now, so you'll be great sugar chemists in the future. All right, so like I said, let me know if you'll be around. So I see somebody will be there around 2, so I'll be there around 2, or we can meet now or whatnot and work on printing or anything else, last minute questions or office hour help. Happy to be there, but like I said, let me know if you're coming so I don't accidentally leave thinking everybody's done. All right, I'll see you all on Thursday then for poster presentations and hopefully a demo. I'll hang around on stream another minute or 2 just in case. Oh, and I forgot to hand back everybody's papers.
You got a copy of the final? I didn't pick one up.
Oh, I can't get one.
Yeah, these are the turn
Yeah, you can make a turn pile. Random. Trying to get out this weird days. Quizzes. Quizzes. Did you blonde your hair?
It's good, but yeah. I bleached it for this.
That looks nice.
Thank you. I'm kind of how it turned out. I was worried I wouldn't like it because I've never done it more natural. I usually do my purples. I think it turned out pretty nice.
Yeah. a little issues with carbon counting on some of those quizzes, but other than that, it's pretty good. Oh, we needed an exam. Let's see.